Histology and its methods of study. Overview. In this topic we will discuss about human anatomy, subdivisions, importance of histology, microscope, microscopy, types of microscope, light and electron microscope, resolving power, magnifying power, source of illumination, objects, lenses, varieties of optical microscope, bright field microscope, preparation of tissue for bright field microscope, fixation, embedding, sectioning, staining, polarizing microscope, phase contrast microscope, dark field microscope, fluorescence microscope, confocal microscope, electron microscope. Types: transmission microscope (TM), scanning microscope, preparation of tissue for microscope, autoradiography, cell and tissue culture, enzyme histochemistry, immunohistochemistry. Human anatomy is a branch of medical science which is concerned with the study of the human body. The two major subdivisions of this subject are macroscopic anatomy, microscopic anatomy. The macroscopic anatomy, more commonly called gross anatomy, consists of the study of the parts, organs, and structure of the human body by the naked eye. The microscopic anatomy, more commonly called histology, is the study of minute structure of the body tissues with the help of optical instruments called microscope. Importance of histology. The knowledge of histology is of paramount importance to understand the subject of physiology, which deals with the study of normal function of the cells, tissue, organs, and organ system. The knowledge of normal histology is also of fundamental importance in understanding the subject of pathology, is the study of changes that occurs in the structure of cells and tissues when they become diseased. Microscope. Microscopes are optical instruments consisting of a combination of lenses, which provide an enlarged image of small objects and reveals the details of structure not distinguishable by unaided eye. Microscopy. The observation by a microscope is called microscopy. The usefulness of any types of microscope depend on its two qualities. Magnification of microscope means its ability to enlarge the image of the object being viewed. Resolution of a microscope implies its ability to allow the observer to distinguish between two small objects. Resolution is inversely related to the wavelength of source of illumination. It means shorter the wavelength, greater the resolution. Now we distinguished a short difference between light and electron microscope. Resolution. Resolution of light microscope is 2 micrometer. Resolution of electron microscope is 2 to 4 angstrom. Magnification. Magnification of light microscope is 500 times. Magnification of electron microscope is 500 to 25,000 times. Source of illumination. Source of illumination of light microscope is light. Source of illumination of electron microscope is beam of electron. Objects. In light microscope living and dead objects are used. In electron microscope dead objects are used. Lenses. In light microscope glass lenses, that is, objective lens, eyepiece, ocular lens, are used. In electron microscope electromagnets are used. Varieties of optical microscope. There are several kinds of optical microscope. Bright field microscope. Bright field microscope generally consist of 5 components: light source, condenser lens, glass slide, an objective lens and an ocular lens. Light source. The light source produces a beam of light which illuminates the tissue specimen which is under observation. Condenser lens. The condenser lens focuses the light on the specimen to be examined. Objective lens. The objective lens gathers the light which has passed through the tissue specimen. This lens magnifies and projects the illuminated image of specimen towards the ocular lens. The ability to gather light and resolve the specimen details depends on the numerical aperture of the lens. For objective lens are available in a microscope, which are fixed on a movable turret located just above the specimen. Out of these, the first 3 objective lenses magnify the image 4, 10, and 40 times. The fourth objective enlarges the image hundreds times and is called oil immersion objective. It is a technique used to enhance the resolving power of light microscope. It involves the immersion of objective lens as well as the specimen in a layer of transparent oil of high refraction index. Ocular lens. The ocular lens, also called eyepiece lens or simply eyepiece, further magnifies the image 10 times and focuses the resulting image on the retina of the viewer's eye. Only one ocular lens is present in the monocular microscopes, whereas the binocular microscopes have 2 ocular lenses. The resolving power of bright field microscope is about 0.2 micrometer. It can magnify the images thousands times to 1,000 times. Preparation of tissue for bright field microscope. Histological sectioning. Most commonly, dead tissues are used for microscopy, which immediately after removed from animal body and treated with chemical preservation and cut into thin slices called histological sectioning. The bright field microscope involves 4 main steps. First process is fixation. Purpose of fixation is preservation of the tissue structure with the last possible alteration. This is achieved by influence of various chemical compounds called fixatives, which render the structure components of the cells insoluble. The most commonly fixatives is formalin. Pure formalin is 37% solution of formaldehyde gas in water. For general purpose, a 10% solution of formalin is employed as a tissue fixative. The major action of the fixatives is to bring about stabilization of proteins, by causing the formation of cross-links between the protein molecules. The change in the condition of protein also results in the inactivation of certain cellular enzymes that would otherwise start the process of self-digestion, that is called autolysis. Second process is embedding. The tissue obtains some degree of firmness by fixation. It is still far too soft to be cut into thin sections. Therefore, prior to sectioning, the tissue must be embedded in a material which, after hardening, has a consistency that permits it to be sectioned together with the tissue piece. Paraffin wax is the most frequently used embedding agent. Paraffin is solid at room temperature but become liquefied when heated to 60 Celsius. The fixed and dehydrated tissue kept immersed in heated paraffin, within small cubical containers which are kept in an oven at 60 Celsius for several hours. This allows paraffin to infiltrate into specimen thoroughly. When the containers are removed from the incubator and allowed to cool, the paraffin hardens to form a firm, cubical block containing the embedded tissue specimen. Third one is sectioning. Sectioning of the tissue block into thin slices is carried out by the help of a machine called microtome. The sectioning are usually 3 to 10 micrometer thick and each tissue section is mounted on a glass slide. The mounted tissue sections are allowed to dry in an oven to remove the moisture and help the sections to adhere to the slides. The dried tissue sections are ready to be stained. Fourth is staining. Most tissues of the body are colorless and as such, it is difficult to study their structure details. The purpose of staining is to enhance the natural contrast, so that distinction can be made between different components of tissue and cells. Staining in general use are considered to be acid or base, but in fact they are neutral salts having both acidic and basic radicals. Basic dye. When the coloring property of dye is in the basic radial of the neutral salts, the stains are called basic dye. The commonly used basic dye includes hematoxylin, toluidine blue, methylene blue. And the structure that stain in basic dye is called basophilic structure. It is important to understand that the basophilic structure which attract the basic dyes are themselves acidic in nature. Example: the nucleus of a cell takes a basic stain because it contains DNA and RNA. By the hematoxylin and eosin staining technique, the basophilic substance are stained light blue to dark blue or purple. Acidic dye. When the coloring property of dye is in the acidic radial of the neutral salts, the stains are called acidic dye. The commonly used acidic dyes includes eosin, acid, fuchsin, orange G, and picric acid. Acidophilic structure. The structure that stained in acidic dye is called acidophilic structure. It is important to understand that the acidophilic structure are themselves basic in nature. For example, the ribosomes of the cytoplasm are basophilic because they contain RNA. The general cytoplasm of the cell stains acidophilic because the constituents protein of the cytosol are rich in basic amino acid. By the hematoxylin and eosin staining technique, acidophilic structure in the tissue take a pinkish to reddish color. In routine histology, hematoxylin and eosin is stain of choice. As indicated by its name, this stain consists of a combination of a basic dye. The staining in general are considered to be acidic or base, but in fact they are neutral salts having both acidic and basic radial. Polarizing microscope. When light passing through a polarizing filter, it comes out vibrating only in one direction. In polarizing microscope, two polarizing filters are used. The first filter is located in below the condenser lens and is known as polarizer. The second filter, known as the analyzer, is placed between the objective and ocular lenses. When the polarizer and analyzer are so disposed that their main axes are perpendicular to one another, no light can pass through, resulting in a dark field effect. However, if substances or structure containing asymmetric molecules which are oriented in a highly ordered manner are located between the polarizer and analyzer, their repetitive molecular structure allows them to rotate the axis of light emerging from the polarizer. Therefore, such things appear as bright structure against a dark background. The capacity to rotate the direction of vibrating of polarized light is called birefringence, double refraction. The substance or structure exhibiting birefringence are called anisotropic. The structure or substances possessing a single refraction are referred to as isotropic. Under the polarizing microscope, the isotropic structure do not appear bright. Phase contrast microscope. This variety of microscope is used to study of unstained fresh tissue or living cells. The phase contrast microscope is based on the fact that light wave passing through region with different refractive indices are deflected differently, and put out of phase with one another. Different cytoplasmic and tissue constituents produce phase difference because they vary in thickness and refractive index. The phase contrast apparatus consists of optical plates placed within the condenser and objective lens, which convert the phase differences into differences of light intensity. Thus, differences in refractive index are rendered directly visible. These are the videos of objects under phase contrast microscope. It is the video of cell division under phase contrast microscope. Dark field microscope. This microscope uses a strong, oblique does not enter objective lens. This is achieved by dark field microscope, in which no light passes through the center of lens. The light thus reaches the object to be viewed at an angle so oblique that none of it normally enters the objective lens. Therefore, the viewer sees the field being examined as a dark background. However, which the specimen to be examined is placed between the condenser and objective lens, the light scattered by some of the structure in the specimen manages to reach the objective lens. Such structure appear bright against a dark background and can be seen easily. The dark field microscopy is employed to render unstained and transparent specimens clearly visible. This is a video of white blood cells attacking on parasites. Fluorescence microscope. In fluorescence microscope, fluorescence compounds having affinity for specific cell components. Fluorescence stain acridine orange bonds with DNA and RNA. In the fluorescence microscope, the treated tissue specimen are irradiated with ultraviolet light, upon which the fluorescence structure emits light having longer wavelength which are in the visible range. The fluorescence microscope contains special filters that selects the rays of specific wavelength, allowing the viewer to see only that which is fluorescence. Confocal microscope. In the ordinary optical microscope, the beam of light is quite large, and consequently the excess light produces a flare which decreases the resolving power of the objective lens. The confocal microscope are specially designed to eliminate the out-of-focus light so that a sharp focus with a high resolution is produced. This microscope employs two major technical improvisations. A small point of high-intensity light obtained from a laser. A plate with a pinhole aperture which is placed in front of the image detector lens. It is the observation of cell division under confocal microscope. Electron microscope, EM. In an electron microscope, a beam of high-velocity electron pass through the tissue specimen, to be examined. The wavelength of electron beam is 1/2000 of the visible light beam. Because the resolution is inversely proportional to the wavelength of the illuminating source used. The resolution of the electron is 2 to 4 angstrom. The resolution of the electron microscope is correspondingly higher than that of the light microscope. The magnifying power of the EM is also greater than that of the light microscope. Its magnification power is from 500 times to 250,000 times. The high resolving and magnifying power of the microscope allows the study of the structure of the body tissue at subcellular level. The structure visible only under the electron microscope is called ultrastructure or fine structure. To be specially mentioned is the fact that quite large areas of a tissue specimen, generally several centimeters square, can be examined under the LM. Whereas in EM the specimen area available for examination is very small, usually being less than 1 millimeter. In addition, the EM shows only black and white images. Whereas a number of staining techniques are available for imparting different colors to various components of the tissue being examined by the LM. Varieties of EM. Two varieties of EM are generally transmission electron microscope, TM. Scanning microscope. Transmission electron microscope, TM. The transmission electron microscope, TM, function on principle that a beam of electron can be deflected by electromagnetic field, in a manner similar to deflection of light in the glass lenses. In this microscope, the illuminating source is a beam of high-velocity electron accelerated in a vacuum. The beam is passed through the tissue specimen to be studied and focused upon a fluorescent screen, or photographic plate by a series of electromagnetic fields. The resolution of transmission electron microscope, TM, is about 2 nanometer. While the ultra-thin tissue sections can be magnified up to 120,000 times. Top emits electrons that travel to an anode with an accelerating voltage between 60 and 120 kV. Electrons passing through a hole in the anode form a beam that is focused electromagnetically by circular electric coils in a manner analogous to the effect of optical lenses on light. The first lens is a condenser focusing electrons reaching the objective lens form an image that is then magnified and finally projected on a fluorescent screen or a charge-coupled device. CCD, monitor and camera. Scanning electron microscope. The scanning electron microscope is used to the view surface of solid specimen. It differs from the TEM in that the electron do not pass through the specimen under examination. The fixed specimen is introduced into an electron beam that scans the surface of the specimen, which has been coated with a very thin layer of a heavy metal, light gold or palladium. Electrons that are reflected or ejected from the heavy metal coat are captured by electron detectors, and finally focused to produce a highly magnified 3D view on high-resolution monitor. The image can also be made permanent either by photographing it or by digitalizing it for storage in a computer. The resolution power of scanning electron microscope is 1 to 20 nanometer. The magnification of scanning electron microscope is 20 to 50 times. Preparation of tissue, EM. For electron microscope the commonly used fixative are glucosaldehyde and osmium tetroxide. After fixatives the tissue is embedded in epoxy resins, araldite. Dehydration. The tissue is dehydrated by gradually exposing it to a series of increasing alcohol concentrations. This process removes water from the tissue to prepare it for embedding. Staining and contrast enhancement. The sections may be stained with heavy metals like uranyl acetate and lead citrate to enhance contrast and highlight cellular structures. The tissue blocks so obtained are subjected to sectioning at machines called ultramicrotomes which produce ultra-thin sectioning ranging from 40 to 100 nanometer in thickness. The ultra-thin sections are stained to enhance the inherent contrast of the tissue cell structures by passing them through a solution of lead citrate and uranyl acetate. Autoradiography. This process localizes cell components synthesized using radioactive precursors by detecting silver grains produced by weakly emitted radiation in a photographic emulsion coating the tissue section, or cells. With either light microscopy or TEM, autoradiography permits Unique studies of processes such as tissue growth, using radioactive DNA precursors, or cellular pathways of macromolecular synthesis. Labeling and sample preparation. Involves labeling molecules of interest with a radioactive isotope. After labeling, the tissue or cells are typically fixed using appropriate fixatives to preserve their structure. Embedding and sectioning. The fixed tissue or cells are embedded in a suitable matrix, paraffin or resin, for sectioning. Thin sections, usually around 5 to 10 micrometers thick, are cut using a microtome. Salivary gland of a mouse injected with 3H-fucose 8 hours before tissue fixation. Fucose was incorporated into oligosaccharides, and the free 3H-fucose was removed during fixation and sectioning of the gland. Black grains of silver from the light-sensitive material coating the specimen are visible over cell regions with secretory granules, and the duct indicating glycoprotein locations. X1500. The same tissue prepared for TEM autoradiography shows silver grains with a coiled or amorphous appearance, again localized mainly over the granules, G, and in the gland lumen, L. X7500. Cell and tissue culture. Cells can be grown in vitro from newly explanted tissues, primary cultures, or as long-established cell lines, and can be examined in the living state by phase contrast light microscopy. Sample preparation. It involves fixing the cells or tissues to preserve their morphology and cellular structures. Common fixatives include formaldehyde or paraformaldehyde. Embedding. After fixation, the cells or tissue samples are often embedded in a solid, paraffin wax or plastic resins, commonly used. Staining. Staining is used to enhance the contrast of cellular structures and components. Stains. Stains include hematoxylin, nuclei, and eosin, cytoplasm. It is used to highlight specific cellular components, such as connective tissues or cell organelles. Enzyme histochemistry. Histochemical, or cytochemical, techniques use specific enzymatic activities in lightly fixed or unfixed tissue sections to produce visible products in the specific enzyme locations. Fixation and paraffin embedding denatures most enzymes, so histochemistry usually uses frozen tissue sectioned with a cryostat. Enzyme classes for which histochemical study is useful include phosphatases, dehydrogenases, and peroxidases, with peroxidase often conjugated to antibodies used in immunohistochemistry. Tissue preparation. The tissue is often fixed using appropriate fixatives to preserve the enzymatic activity and cellular morphology. Embedding and sectioning. The fixed tissue is embedded in a suitable matrix, paraffin or resin, to facilitate sectioning. Thin sections, typically around 5 to 10 micrometers thick, are cut using a microtome. It is the picture of enzyme stains. Micrograph of cross-sections of kidney tubules treated histochemically to demonstrate alkaline phosphatases, with maximum activity at an alkaline pH, showing strong activity of this enzyme at the apical surfaces of the cells at the lumens, L, of the tubules. X200. TEM image of a kidney cell in which acid phosphatase has been localized histochemically in 3 lysosomes, Li, near the nucleus, N. The dark material within these structures is lead phosphate that precipitated in places with acid phosphatase activity. X25000. Visualizing specific molecules. Based on specific reactions between an antigen and antibodies, often fluorescent compounds or peroxidase for light microscopy, gold particles for TEM. Direct immunohistochemistry. If the cell or tissue antigen of interest is detected by directly binding a labeled primary antibody specific for that antigen, the process is considered direct immunohistochemistry. Indirect immunohistochemistry uses an unlabeled primary antibody that is detected bound to its antigen with labeled secondary antibodies. The indirect immunohistochemical method is more commonly used because the added level of antibody binding amplifies the signal detected and provides greater technical flexibility. In situ hybridization, ISH. Specific gene sequences or mRNAs of cells can be detected Microscopically using labeled cDNA probes in a technique called in situ hybridization, ISH, a single cultured uterine cell stained fluorescently to reveal a meshwork of intermediate filaments, green, throughout the cytoplasm. Primary antibodies against the filament protein desmin and fluorescent isothiocyanate, FITC. Labeled secondary antibodies were used in the indirect staining technique, with the nucleus counterstained blue with DARPI. X650, small intestine treated with an antibody against the enzyme lysozyme. The secondary antibody labeled with peroxidase was then applied, and the localized brown color produced histochemically with the peroxidase substrate 3,3-diamino-azobenzidine, DAB. The method demonstrates lysozyme-containing structures in scattered macrophages and in the large clusters of cells. Nuclei were counterstained with hematoxylin. X100, a section of pancreatic cells in a TEM preparation incubated with an antibody against the enzyme amylase, and then with protein A coupled with gold particles. Protein A has high affinity toward antibody molecules, and the resulting image reveals the presence of amylase with the gold particles localized as very small black dots over dense secretory granules and developing granules, left. With specificity for immunoglobulin molecules, labeled protein A can be used to localize any primary antibody. X5000, specific antigens with diagnostic importance. Antigens. Diagnosis. Specific cytokeratins. Tumors of epithelial origin protein and polypeptide. Protein and polypeptide hormones. Certain endocrine tumors. Carcinoembryonic antigen, CA. Glandular tumors, mainly of the digestive tract and breast. Steroid hormone receptors. Breast duct cell tumors. Antigens produced by viruses. Specific virus infections.